I'm Dr. Susana Puig. I'm the director of the dermatology department at the hospital clinic in Barcelona. I'm also the chief of the research group at EDBABS that it's called Melanoma Imaging Genetics and Immunology, and the senior author of this study in which we analyze POT1 and promoter TERT mutations in families with familial melanoma.
And I'm Potrony. I'm a PhD at Dr. Susana Puig's lab, and I'm the first author of this article. Around 10% of melanoma cases occur in a familial context, and we know that today CDKN2A is the main gene involved in genetic susceptibility in familial melanoma. And in Spain, we know that 14% of families have mutation in this gene, but we still don't know the mutations in other genes in our set.
For this reason, it's extremely important to identify new genes that we can offer for genetic counseling. And this is important because we can identify healthy individuals that can carry this risk for melanoma and that can be followed up in a special setting to perform an early diagnosis of melanoma.
In this study, we sequenced POT1 and TERT promoter gene in 228 families with unknown mutations in high-risk genes for melanoma susceptibility, and we combined high-throughput sequencing technologies with classic Sanger sequencing. For those families in which we identified a likely pathogenic mutation, we performed co-segregation analysis, and also for those mutations that could affect the splicing, we performed functional analysis of the mRNA.
We have identified POT1 mutations in 2% of the families that we studied. On the contrary, we didn't identify any mutations in the promoter of TERT in any of the families. This is important because in families with POT1 mutations, we identify other types of cancer, as thyroid cancer, not only melanoma. And at the time that we are doing genetic counseling and we are following our patients, we can offer them early diagnosis not only for melanoma but also for these other types of cancer.